everyone. Welcome back for our final section of sessions for the day, where we're going to hear about the future of digital health, innovation, AI, and what's next. And I'm thrilled to welcome on stage my good friend, Dr. Daniel Karlin, Chief Medical Officer of MindMed, also the chairman of the board of DIME, and an entrepreneur himself. Welcome, Dan. And a DJ.
Oh, do I get my own?
Yes, you get your own.
Thank you for that rather anemic applause.
Nobody likes it.
Did anybody else download Grindr after that last one? Re-download Grindr? Okay, so I'm gonna have to tell you a story kind of quickly for any of this to make sense, because I think often when we go looking for simple solutions, we find that the world is far more complex than we wish it were. And I think what I'm gonna be able to tell you about is an application of large language models that actually make something better in clinical trials. And that's really ought to be the goal, right? That so often I feel like we end up chasing problems with a solution, that there's some solution or something that seems like it ought to be a solution, some sort of new technology that appears in the world, and suddenly it has to be applied to everything, even when it doesn't help, even when there's not a real problem that's been identified. We're just gonna spend the next 15 minutes quietly reading this to ourselves. Suffice to say, I may be making forward-looking statements and don't trade on it or anything. Obviously, I can't tell you anything that we don't say publicly, but SEC and stuff. Is there still an SEC? Is anyone up to date on that? One can reasonably assume at some point there might be another one, so we should still do the right thing in general. MindMed's a small biotech that is doing essentially big pharma stuff with many fewer people, and the conclusion I've come to, having done clinical trials both in a very large company and in a very small company, is that there's almost nothing that you can do with 30 people that you can't do worse, slower, and more expensively with 300 people. And so we've got some staff here in the room, but effectively with a team of about 30 in a clinical group, we're running 3 phase 3 clinical studies in GAD and MDD. It shouldn't be lost on anybody here that GAD and MDD represent massive burden and suffering in our community. Folks are fond of citing the statistic that 1 out of 5 people suffers from mental illness, but who are we kidding? Have you looked at the world recently? Like 5 out of 5 of us are anxious. 5 out of 5 of us have intermittent depression. I'm a practicing psychiatrist, I'm a psychotherapist. One interesting thing that emerges in psychiatric trials is that almost inevitably you run into more difficulty with your so-called healthy volunteers than people who at least walk into the clinic admitting that they don't feel very well. Be very cautious around people who go out of their way to tell you how good they are. It's called narcissism. This is the intimate, small afternoon crowd. We can say stuff we couldn't say when all the rest of them were here. You guys are special. So we're gonna be talking about LSD, and so between Grindr and LSD, we're really setting ourselves up for a hell of a weekend, I guess. How does LSD work to alleviate distress? How does it work to alleviate MDD and GAD? I could tell you some theories, and I'm about to, but the reality is we don't know. The same way we don't know how SRIs work. We don't know how GLP-1s work. In general, we have to be very comfortable living in a world where we don't really know how we work, particularly when we talk about diseases of the entire system, of the whole human body. And there's no way around the fact that diseases of the brain are diseases of the whole human body. It's a weird little delineation thing we do where basically psychiatry has been willing to say, if you can see it on a scan or detect it in the electricity, then it doesn't have to be psychiatry anymore. It gets to be somebody else's problem, right? So we end up with anorexia being a psychiatric disorder, but all of these people getting GLP-1s for eating too much, no, that's primary care, that's cardiometabolic, right? And totally arbitrary where we draw these lines. So arguably GLP-1s are another brain drug that we don't really know how they work. What we do know is that when people in a therapeutic setting, and we know it's true in therapeutic settings because we bother to measure people in therapeutic settings. We're not running around to Phish concerts and trying to evaluate this, but that when people take LSD at a fairly high dose in a therapeutic setting, the normal organizing principles that they use to understand the world go away. These drugs have been termed hallucinogens. That's just a misnomer. They don't cause hallucinations. What they do is they remove the way that you normally understand the world, the ways that you normally see the world, and the ways that you normally hear or taste the world, right? You hear about sensory crossing, where you might hear a sound. Nope, you're supposed to hear sounds. Hear a color. No, nothing? And built into all of that perceptual change is a loss of language. Arguably, your distress at existing in the world as you experience it is built of language. And your anxiety about the future is based on linguistic constructs. Your depression, if you're feeling down, is based on linguistic constructs. When those things go away, when people are in the clinic for the purpose of feeling better, what seems to happen is that they are able to reevaluate and reconstruct those filters as they emerge from the experience in such a way that their relationship to themselves, their relationship to the world, is different when they emerge. There's also probably neuroplasticity. When people talk about neuroplasticity, remember, when it comes to neuroscience, causation is complicated. And so neuroplasticity is definitely either the cause of or a measure of the change. Because you can't have change in the brain without neuroplasticity. So why we call it cause in one case and measure of change in another, I don't know. But be suspicious of too much reliance on claims of neuroplasticity. And if you're listening to me now and you remember any of it, that's great. We're doing neuroplasticity. That's the only way, I hope that's the only way you can remember stuff. Otherwise, I don't know where you're storing it. So what we did was a phase 2b study to look at dose effect of MM120, which is our pharmaceutically optimized form of LSD. 5-arm study. I got to tell you about this so I can tell you about the phase 3, so I can tell you about the AI. We're going to speed it up now. And what we saw is that at 100 micrograms, which was our best dose, a very substantial placebo effect that was very substantially exceeded by a drug effect on both the HAM-A, which is the regulatory endpoint for GAD, and the MADRS, which is the regulatory endpoint for MDD. That gave us really strong rationale to advance this drug into pivotal clinical studies in both GAD and MDD. And what you see here, I think, is important to note, which is that not only are we able to elicit a statistically significant change in these studies, we're able to elicit a real clinically meaningful change. People going from, on average, severe disease to either remission or mild disease. These are changes that matter to people. And again, it's a massive absolute change, almost 23 points, that's beating a nearly 15-point placebo. When you hear people complain about placebo effect, it's nice to remind them that placebo doesn't cause placebo effect. Participation in the trial causes placebo effect. Therefore, both arms had the benefit of the placebo effect. And if your drug doesn't exceed placebo, it's not placebo's fault. That said, I have knowingly prescribed placebo in my clinical practice. I really don't like the drugs we have available to treat people today. Low adverse event burden, almost entirely contained to the day of dosing. Again, all of this good rationale to bring forward a dose. We evaluate our primary outcome measures using centralized raters, and this becomes important for the digital thing I'm about to tell you about. What does a centralized rater do? They run through a ClinRO, a clinician-assessed outcome measure, with the participant to get at their level of disease. Those the HAM-A and the MADRS, both ClinROs, versus, say, the PHQ-9, which is a patient-reported outcome measure, right? We think that these are more reliable. Certainly, the regulators are much more prone to accept ClinROs than patient-reported outcomes. We designed a phase 3 pivotal program for both GAD and MDD that largely parallels that phase 2 program and its primary outcome, so a 12-week double-blind observation period. And at the end of that 12-week, we assess our primary outcome measure. Skip some of these details. So again, we're taking this into GAD and MDD in independent programs. So here's the HAM-A. 14 questions, scoring from 0 to 4 on a per question basis. interviewers ask a structured interview guide that has been validated for good inter-rater reliability at assessing people's level of anxiety. What's the problem? It's hard to get at this, right? You're trying to translate someone's entire experience of the world, their experience of themselves, into ultimately an integer. You use a bunch of sub-integers to try to get it in rate reliability and get it symptom clusters and to structure the thing so that it's more reliable both within a patient and between patients. But ultimately, a lot of variability can be introduced through this rating system. It's why people prefer things that can be directly measured, right? We like to measure things that are easy to measure, even if they're not that important to measure. And it's funny because we tend to poo-poo measures like this while holding in often too high regard measures like blood pressure, even though we know that in-office blood pressure has little relationship to walking around blood pressure, right? But easy to measure, nice numbers. This number's harder to get to. So what we did was use ultimately a large language model that is able to listen to and evaluate the conduct of that rating session so that as the central rater conducts the interview, the model listens to the questions being asked, listens to the responses, and ultimately encodes those as data that it can evaluate against a score. We trained this on our phase 2 data. We obviously had a lot of sessions from the phase 2, and ultimately came up with a model that when tested on a holdback set of phase 2 data is able to show extraordinarily high inter-rater reliability with the average rater. The main point here being that we got to treat something as ground truth. We're not trying to create a predictive model. We're just trying to create a concurrent criterion validation model that says the right rater, the rightest rater, or the average consensus of raters, the model will tell us what they would say. The goal here to detect outliers. So as we bring this model into phase 3, it ultimately allows us to evaluate the HAMI score. Todd gets to laugh because he named it, but the HAMI score against the standardized rater score. And what we see is, again, high level of alignment between the model and the professional rater, both at high levels of severity and lower levels of severity. And this is sort of what you can see across a single interview. And this is an interesting one that was selected for this deck because this is a divergent case where the HAMI would have rated the person differently than the initial rater did. And we see one of the pitfalls of misrating, which is that if a baseline rating isn't done as well as it could be, a subsequent better rating might more accurately capture the person's condition. And in this case, it appears that the rater overrated the subject at baseline, and therefore we end up calling that person a placebo responder because a more accurate rating captures that they aren't as sick as they seemed. And this is what we get to do in phase 3, which is to, in near real time, evaluate the performance, all of the raters working on the study so that where we observe outliers, one, we can just click on it and go look at the text and say, does this make sense? Is the model picking up something that is divergent or not? So we can do 2 whats? Okay, 2 minutes apparently. I don't know what else it could have been. I'm teasing Lauren because she's had a long day. And it lets us, in near real time, evaluate the quality of the most important information that comes out of the study, which is the measure that is the reason for the existence of the study, and ultimately the measure that will allow us to go to FDA and try to get a label for the drug. And I think this is just more examples of the same, and why not leave it here so we can get a question or two? Or do we want to move on because we're running a little late?
We have time for one question.
One short question. One long question, Joe. Whatever you want.
All right, this is wild. Wait, Joe. So what you're doing, I don't know if anyone here.
Doug's got the mic.
All right, so this is wild. What you're doing is you are now taking what is considered one of the most expensive pieces of services in clinical technology, which is raters and central review, in turn training an AI to replicate what they do. Now that's going to take a lot of time. So when do you have to train these on indication-specific use cases in
Scale specific.
Scale specific.
Yeah.
Okay.
And it's done. We now have an automated HAM-A large language model that can conduct the HAM-A as well as a rater. I don't want to have that fight with FDA. We'll keep paying for raters for now, but the thing works, and it works on its own, so it can both evaluate rater performance on a per question basis, but it can also conduct the interview on a per question basis.
So have you had regulatory feedback now that they've embraced AI now yet at the FDA?
Now, like today,
I mean,
yeah. I mean, literally there was an article this morning that they're implementing the AI faster than expected. We'll talk to them about this, but this, I mean, you've heard me say this before. You get to do like one weird thing per clinical trial. And we're using this for our own good to enhance the integrity of the trial for our sake. The weird thing that we're doing in our clinical trials is the acid part. And that's enough. I'm not trying to, that's why we're using a nice traditional outcome measure, because it's trying to wrap something that seems unusual in as much stuff that seems really normal as possible, admitting that in a world where FDA is being directed to implement AI to do what it's not clear, nothing is totally normal in today's world.
So what have you done with the results of what you've seen? Has it caused you to pivot and change because you're getting insights from the LLM that are helping you revalidate your data? How are you using
so the data are the data. And as a sponsor, we obviously have a distance from those data and the ability to change them. But it's about enforcing integrity as you move through the study. So one divergent rating is not something we would normally be able to pick up with really anything that exists today except for this. But after even one divergent rating, we can go talk to the rater. Not because we can go back and change that. You're stuck with what you measured, but because we don't want another one.
Right, thank you, Dan.